All right, we'll finish these lecture slides. That'll complete chapter 3. And we have a DNA lab today. The DNA lab, well, I'll explain that after break. Well, let's start with the lecture material from genes, proteins, or protein synthesis. From nucleotides to proteins. DNA, nucleotides, carries instructions on how to make proteins. And it's a one gene, one protein relationship. One gene, which is the DNA, equals one protein. And as you'll see today, you just link together amino acids to make a polypeptide chain. One gene, one protein. That's the first thing to understand. The genetic information is conveyed by a linear sequences of the 4 bases, A, T, C, G. Okay, so that's what a gene is. Just a certain order of bases. And what happens is, well, understand that the gene and its polypeptide products are collinear. Gene is the collection of adjacent nucleotides specifying the amino acid sequences for one polypeptide chain. The corresponding nucleotide and amino acid sequences are collinear. So what they're just trying to say is, just forget about DNA for a second. Let's pretend you're coding a message, and your code is the alphabet. You code with numbers. So in your secret code, A, the code for A is number 1, and B is number 2, and so forth. What's the code for Z? 26. In your code that's impossible to decrypt. So that's your code. Now, let's say, I don't know, you want to convey, so if your code was like, so I put some random numbers, and if I want to figure out what letters I'm trying to, if I want to decode that, let's see, 1 is A, C, D, B. Oh, I decoded it. And what I did was, I just went from one way to the other in a line. This was collinear to that. That's all they're saying. The DNA nucleotides, you just read it in a line. And then you get your polypeptide chain in a row with it. Okay, you don't skip around. You just read it. You transcribe the gene, just like I did, from left to right. Now, okay, let's get back to nucleotides and amino acids. You see on the left there? Count. How many nucleotides are there? How many nucleotide bases? 1, 2, 3, 4. 4 nucleotide bases. The A, the T, the C, the G. 4. 1, 2, 3, 4. How many amino acids are in that picture? There's 20. Amino acids are what you're building. What I'm telling you is, the nucleotides code for the amino acids. Do you see a problem? Okay, let me see if you're thinking what I'm thinking. Let's say the relationship was 1-to-1. One nucleotide can code for one amino acid, just like 1 was my code for the letter A. If that's the case, which it's not, but let's say it is, if it's a 1-to-1 code, how many amino acids could you code for? You could code for 4. But what about the other 16? Okay, so you see that's the problem. So what they understand is the correspondence between nucleotides and amino acids is not 1-to-1. If it was 1-to-1, you would need 20 of these to code for 20 of those. So that's not how the code works. Okay, well, it's mRNA that's used to make the codon assignments. This genetic code was fully deciphered by 1966. And what they figured out was it's not 1-to-1. They put these in triplets, okay? And it's not DNA, it's RNA. And let's remember that RNA has no thymine. So when it's RNA, there's no thymine. And so what they figured out was this relationship was mRNA codons. So think of it in sets of 3. And you're going to have in this line. Each code or codon are 3 nucleotides. So you have 4 bases. You put them in sets of 3. That can actually give you like 64 permutations. And you only needed 20, so you have some redundancy. Amino acids are specified by more than one codon. So what that means is, for example, the amino acids, we didn't learn their names, but sometimes they have 3-letter abbreviations, like LEU is leucine, that's proline, that's arginine, serine, arginine, and if you take biochemistry, you would understand that. But for us, just understand that that's a 3-letter abbreviation for one amino acid. So for example, CUU can code for leucine. Okay, that triplet, that can code for leucine, LEU. Okay, this is your mRNA. And you decode it for your amino acid. But what else can code for leucine? CUC, CUA, CUG. Any of those can code for leucine. Okay, so that's kind of like the explanation. And of course, to make a protein, you need to start and you need to stop. And so there are signals for that. AUG is the code for the amino acid methionine, MET, but that's just the start signal. So that's the codon. And you code for methionine. And that's start. Okay. So you start and you start linking the amino acids together. And at some point you have to stop when you're done. And so there's a few codons for stop. So you can see what they are there. So you are expected to know how to use this table. For example, so we'll get to that. First of all, we'll use this table throughout the lecture, but that's the code that they came up with in 1966. They figured out these mRNA codons code for the amino acids to make these different proteins, and they figured out how it starts and they figure out how it stops. Okay, so one thing is that you have the degeneracy of the code because you have more than one codon that could code for one amino acid, so that's the degeneracy of the code. So we have more than enough. We only needed 20. We only needed to code for 20. You have many more codons that could code for those 20. We're well covered. Now to go through the mechanics, we know what the code is. How does it actually work? How do cells actually accomplish this? Let me remind you of a few things we talked about last time. Okay, read that question, multiple choice. 2 choices. Where are proteins made? Nucleus or cytoplasm? Where do you actually link the amino acids together in your cell? Nucleus or cytoplasm? What we had said was all the machinery is in the cytoplasm. The other thing I did mention, I want to make it clear now, does DNA ever leave the nucleus? When you make proteins, the answer is no. The answer is no. Okay, so where I'm going with this is if proteins are made here in the cytoplasm, the DNA is here and it never leaves there. You need some go-between molecule to make your protein in the cytoplasm, and that is, as I've been saying, that's the job of the RNA. Now the RNA is needed because DNA, it needs an intermediate molecule to decode the genetic message, and it needs to be a messenger of the genetic message. That's going to be our RNA, and that's what RNA does. It decodes and carries information to the cytoplasm for protein synthesis. As the key. Quote unquote, it decodes the DNA. That process is called transcription. We're transcribing the gene. It also acts as a messenger, okay? It decodes and then carries the information. It's acting as a messenger. It's called messenger RNA. So I'm writing, it's carrying the code to the cytoplasm. So then you have to actually make the protein. The genetic information, the nucleotide sequence, which is coded by the RNA, which is the mRNA, it would then move into the cytoplasm where it orders up the amino acids. And so that you're actually translating the code. That's where you're actually going from the mRNA to the amino acids. The code in mRNA is translated by linking together the correct amino acids. By linking together amino acids in the correct sequence. So the 2 key words here are, number 1, transcription. You transcribe the DNA. You do that by linking together mRNA nucleotides. And then so you go from DNA to mRNA. And that's your messenger. It's a molecule that physically goes from the nucleus to the cytoplasm. And in the cytoplasm, the mRNA is then translated That's the key word here. It's called the process of translation. That's why I use that word. We order up the amino acids. So that's the last thing I'll double underline. So what we've done here, since I've highlighted my key words, you've gone from your genetic code to the DNA, transcribe it, connect the dots here. And you get your RNA, and then the RNA, mRNA is then translated, and you go from mRNA to amino acids. So you should train your mind. This is how cells work: DNA, RNA to protein. And it's really interesting to understand how important RNA is. You know, how did they know RNA? Well, what were they thinking, that it's RNA that participates in the decoding of DNA? What they notice is that cytoplasm of cells that make lots of proteins had lots of RNA, so there's an association there. Also, DNA and RNA are very similar. The only difference is, instead of thymine, you have uracil. I've been saying that, okay? All right, well, anyways, another similarity. Basically, RNA is a single-stranded version of the double-stranded DNA, so they're very similar. And, the thing is, this is what we came up with, right? DNA to RNA, well, specifically mRNA, and then amino acids or proteins. That's kind of why I did that for you. The process of transcription to the process of translation, this is known as the essential dogma of molecular biology, okay? They figured out that the genes are in the DNA, and the biochemists have even figured out, well, okay, well, enzymatically we figured out how it works. And this is that picture I showed you before, I believe, where you go from DNA to mRNA. Get that pointer. I'm leaving out a lot of details. When you get your mRNA, there's some processing of the RNA. We don't need to talk about that. But you need a way to get the code out of the protection of the nuclear envelope to the cytoplasm. So this ribbon represents the code because you've base paired with it. So you've got the code in the order of your base pairs. And then those base pairs you've translated into your amino acids. This little chain of beads is the end result here. In today's lab, we're actually going to do a DNA isolation using strawberries. And the strawberries, it's not hard to isolate the DNA. All you got to do is mash a strawberry. Okay. Now, do you see two lipid membranes you got to get rid of? That's made out of lipid, and so is that. So what you can do is, after you mash, you can kind of like mix in some detergent, soap, because soap will help separate the DNA from these lipid membranes. But do you remember the structure of chromosomes, which have the DNA? There's also histone proteins in there. And so our detergent has salt with it. And the salt will separate the DNA from the histone proteins. Okay? And that's one of the lab questions I ask you. Well, why do you have salt in the detergent? Once again, soap separates the lipid from the DNA. The salt separates the protein from the DNA in your DNA isolation. So you'll be doing that today in lab. All right, well, anyways, back to protein synthesis. There are actually the 3 RNAs. mRNA is like the messenger one, but there are other functions for the RNA. Okay, and back then, before it was clear what was what, they just knew that there was 3 different kinds. And I just call it RNA1, RNA2, RNA3. And they could kind of quantitate in a given cell or cells. RNA1, that seemed to be the most, 80%. RNA2, 15%. RNA3, something like 2 to 5%. So remember, you know the 3 RNAs. There's no news there. mRNA. What are the other 2? got a T. Very good. There's the third one. rRNA. Now, cellular RNA, just in quantities, it goes 80%, something like 15%, something like 5%. As it turns out, well, for example, I keep talking about this one, the all-important messenger. You're making proteins, you have lots of RNA, but you have to take an educated guess if you didn't look at the slide yet. Which one do you think it is? You know, most students think it's this one, if you just had to make an educated guess. Turns out, it's this one. Even though it's the least amount, it has the most important function. A little matching here. Turns out, the ribosomal RNA is the most, quantitatively the most. It has a large and a small subunit. And the transfer RNA, which transfers those amino acids, is 15%. Okay, yeah?
Ribosomal RNA be related to the ribosomes then, which are the proteins?
Yes. One and the same. Yeah, pretty much one and the same. So I just kind of tell you there, the ribosomal RNA, they're basically the protein factories. We'll see nice pictures of them in your book. That's the 80%. The transfer RNA is 15%. And the messenger RNA, even though it's the least, it has the most important function. It's carrying the protein building instructions. It has a template role on how to do that. It's basically a half DNA molecule. All right, so the three classes of RNAs were known. Which one carried the template of DNA from nucleus to cytoplasm? It's the RNA. All right, so now I'm going to kind of get with the pictures in your book. Now, they're really hard for me to teach from. They're more for you to study. I'll do my best here. But what you're seeing here are all the RNAs in action. So let me kind of just point what you should be seeing here. If you look inside the nucleus, that's where transcription occurs. Right there. You gotta open up the double helix. Okay? You gotta open it up. So that way it's double-stranded. You expose the strands, okay? And then they call them different things. One's the coding, one's the template strand. We'll go through it. And then the mRNA will base pair with one strand, and then it'll leave the protection of the nucleus, go to the cytoplasm. Here's your mRNA transcript. There's a nice little picture you can see. And here's the small and large subunit of the rRNA. And what you do is you just read it collinear, one direction. And as you do, the transfer RNA will transfer the correct amino acid based upon its base pairing with the mRNA. So it gets a little confusing. I'm going to walk you through it. There's a lot of base pairing going on here. You base pair with the DNA to get to the RNA. And you base pair again, tRNA to mRNA. So there's a lot of base pairing going on. And I always like to walk students through it. And I'm not going to really teach the enzymes, but I do want to mention the role of enzymes to help all this chemistry in the cell happen. Because in 1953, that's when Watson and Crick sent their paper to Nature, and they were right, and so that was when the 50-year quest was over with the discovery of the double helix. So for biochemists, their role was just beginning, because now you have to explain chemistry-wise in a cell how transcription, translation will even work. For transcription, you need enzymes. I'll just mention just a couple. The enzymes in transcription are known as the RNA polymerases. Just like you needed a couple enzymes to know for DNA replication, you also need to know just a couple for transcription, translation. So there's one to know. Enzymes that make RNA are known as RNA polymerases. And I'll give you one condition to know. I think it's pretty obvious. If you're going to make RNA, the DNA has to be there, right? Otherwise, there's no code. There's nothing to decode. So here's a simple picture of it. These are the steps of transcription. Initiation. In initiation, nothing's happening yet. Okay, but what you see is that there's a promoter region, and when, with the help of other things like transcription factors, you don't need to know that, but what you see here is the mRNA will start transcribing the gene at the promoter region. That's basically the start of the gene. So I'm writing promoter region is the start of the gene. And you're going to move in one direction, and as you elongate, what you're doing is the enzyme is opening up double strand and reading in one direction. Elongation, as the RNA polymerase moves along the template strand, elongating mRNA transcript one base at a time. It unwinds the DNA double helix before it rewinds the double helix behind it. I think it's good for you to see this illustration where they don't show the DNA as a wound helix. They just show it as a straight ladder. Today, you're going to be building DNA, and it's just a bunch of little balls and springs. And I got some butcher paper out there, and you're just going to lay it out, and I'll show you again right before lab. You're just going to build a ladder representing DNA. Sugar phosphate backbone, make those a couple of colors. And you got your 4 bases, the A, T, C, G, and you'll link them up and just make a ladder. And then I can come and check you off. And you can even, like, because it's all springy, you can like twist it. There are new kits, and I haven't used it before. This is the first time. You guys are my guinea pigs. But people tell me it's fun. So I expect you guys to have fun when you build your DNA. All right. But what we're talking about here is elongation. I'll just say elongation of the mRNA transcript. That's basically what's going on. They call it a transcript. It's just base pairing with the DNA, and you're getting your nucleotide sequence of the mRNA. So in this picture, the transcript is simply this blue line representing the nucleotides of the mRNA. And it's got to be the correct sequence because you're base pairing with one of these strands. In termination, you just break apart. The mRNA synthesis ends with the termination signal being reached. The RNA polymerase and the completed mRNA transcript are released. Termination. Releases mRNA and the mRNA and the RNA polymerase. So not a lot of details there, and here's kind of where I want you to understand the details. I want you to be proficient in base pairing, okay? So DNA is double-stranded. Let's say I give you half of a code. So DNA, double-stranded. Here's my DNA. It's double-stranded. One strand is the coding strand. The other strand, you call it the template strand, and that's the one you actually base pair with. And you transcribe it. But let's just start with, well, I give you one of the strands. You should be able to base pair to get the other strand. So if the coding strand is, let's say we just take a big chunk out of the middle, and ATG, CCC, GAT, TGA. And I'm telling you, okay, well, given that's the coding strand, I ask you on the slide, what's the template strand that goes with it? And I always have to practice this myself every year when I teach this. Even I make mistakes, you have to be careful. What does A pair with?
T.
So you guys say T? T is correct. So then A and T, and then it goes A. And what is G?
C.
Yeah, C, that's correct. So then GGG. And C. And you guys can practice this in your notes. A, C, T. So that would be, that's how you base pair. And if you know one half, you can figure out the other half, right? That's what I said in lecture last time. That's the template strand. So this is our DNA, right? This is our D, and I just gave you half of the strand to make sure you could code it. Now I'm saying, well, okay, if you're going to transcribe, what you do is you base pair with the template strands. Now I'm saying, what's the mRNA codon? Well, what you would have to do is base pair with the template strand. So for mRNA, codon, A, T goes with A. Remember, A, if there's no T, so that's where you put the U. And that's where I always get confused. I always like when I put a U there, but it's not, it's A. You substitute, remember for mRNA, it's U, not T. And even I will write this just to remind myself, just put U, not the T. What goes with C? It's still G. What goes here? C, C, C. That's that one. C, G, A, U. This is T, G.
It's you.
Oh, I'm sorry. See, I make that mistake again. Even I make a mistake. That's why I always write that. I would have caught it. I would have caught it, yeah. All right, so something, look at the codon and look at the coding strand. What do you see as a similarity or a difference? Let's see here. They're the same, except what's the difference? Yeah, it's U, not T. That's why this is called the coding strand, because when you base pair with the template strand, you get the coding strand. That's what you're going to decode when you translate. All right, so we did that. Any questions on the base pairing thing? Again, always check yourself. Even I make mistakes. You will be expected to do that on your exam. Now, here's kind of a nice picture that kind of shows you the whole thing, where you go from template strand at the top in that pink color. When you transcribe, you're base pairing each triplet with RNA nucleotides. However, there's another level. To bring the correct amino acid to build your protein, there's an anticodon for the tRNA. Okay, where A goes with U, U goes with A. It's still CG, right? CG, CG. And you got to do that too. So we'll take it another step. Let me move on here. Oh, okay, this is a different picture showing the same thing. I think I just gave you two different figures of the same figure, just in different editions of the book. So, again, template, mRNA, tRNA, codon, anticodon. The confusing thing is, when you use this table, I will always make it so that this is the mRNA. So when you decode, look at your mRNA, okay? But I want to make sure that you just know how to use this table. So I want to give you a couple of minutes. Here are your mRNA codons. Can you figure out which amino acid each of those codes for? I'll give you a second. Go ahead and do it now. And I'll check back in just a minute. And don't worry, you won't have to memorize this table. If I expect you to use it, I'll just give you the table on the test. See, so AAA. Okay, my first nucleotide, my first base is A. My second base is A. My third base is A. AAA. Lysine, L-Y-S. Okay, did you get that? Let's do the second one. You tell me what you get for GUU. Oh, right there, valine, okay. All right, so any questions on how to use the table? I expect you know how to use it. I'll give it to you on the test. There are different tables. They make them so you read the DNA, but I'm just going to make it simple. You do it from this level, the mRNA. Let's base pair to go from mRNA to tRNA, the anticodon thing. So on the previous slide, they showed the anticodon base pairing with the codon. So now it's all RNA to RNA. So let's make another level. Let's just do this. Here's tRNA and codon. Okay, what do I do here? Let's see, that goes U, A, C. G, G, G. C, A, U, A, C, U. Okay, that's my anticodon. But the thing is, this tRNA has the important job of transferring the correct amino acid. So what I got to do, I got to draw an amino acid associated with it. Okay, and again, always read from the mRNA level. So let's go ahead and do it using our table. Did I mess one up again?
Yeah.
Oh gosh. Bam. All right, so to use the table, always read from the mRNA level. What's AUG code for? Methionine, start. Common mistake here is you always want to read from the last one you did. But you don't read GGG. What do you read? CCC. So always circle it. Proline, PRL. Okay, GAU. Aspartate. ASP. This one. UGA. Stop. Okay. So there's nothing. Stop is not an amino acid. That's just stop. But pretend you have a 3 amino acid polypeptide chain, methionine, proline, aspartate. They're all linked together by peptide bonds. Okay, so that's kind of what we did there. So that's this next slide. We did it. If I give you a coding strand, you should be able to base pair to get the template strand. Then you should be able to base pair to get the codon, and then the anticodon, and then figure out what the amino acid sequence is. Some more details about translation. In addition to base pairing, you should know that for translation, when you went from mRNA to tRNA to get your polypeptide sequence, the rRNA is not part of this, but that's kind of where the 3 RNAs come together. There are a couple of subunits, a small and a large, and then there's mRNA and tRNA with the anticodon sequence. First of all, any questions on this before I erase it? You know, you'll have a chance to work on this in your DNA lab. I think I made that the last thing. That's one of the things I updated. Okay. Let's look at the transfer RNA in detail. All right. Basically know that the anticodon is what you base pair with from the mRNA, but that's the important thing. That's why this is a transfer RNA. You're carrying the correct amino acid to the site of translation. Okay. That's kind of what you see here. There's a lot of steps going on. So in this first thing here, let me see if you understand the figure. This big thing here, which of the 3 RNAs is this one? With the EPA site. It's the rRNA. This long ribbon transcript, which RNA is that? The mRNA. What about this little one here? That's the transfer RNA. Do you see how they all come together in the cytoplasm for translation? That's kind of basically what you see from this picture. Translation occurs in the cytoplasm. You have these 3 RNAs in action. They're working together. Your book goes into detail about the EPA sites, and I'm not going to test you on that, but I will mention in today's lecture since it's such a nice picture. What you see, they're in the middle of it, okay? And you can see your beads here. That's probably methionine at the very start. But as you link them together, this middle site, you can see they base paired codon to anticodon. And then in the A site, the incoming, the one you're going to put on next, leucine, GAU matches with CUA, okay? That's what they show you here, okay? Before and after, and okay, now you're ready. What they've done is, now the leucine is a part of the chain. Before, it was proline. When the leucine came in, now they linked it, and then it shifts in one direction, okay? They call that a translocation, the direction of the ribosome unit. Now it's back to the P site, okay? And you eject the previous one, and another one will come. If it's the end, a release factor will occur. What I expect you to know of this is basically what we already did. Just base pair the codon with the anticodon and know how to read the table, okay? Know which amino acid it will code for based on the mRNA. That's what I expect you to know. When it terminates, the thing you got to know is this polypeptide, it needs to fold and take shape. When translation ends, the polypeptide chain must be processed. By the ER, the endoplasmic reticulum, which we talked about before. There's a nice picture of many rRNAs making their polypeptides. So what happens to them? Well, some just enter the cytoplasm, many enter the endoplasmic reticulum and move through the endomembrane system where they're modified and packaged. The newly forming polypeptide, it may contain a signal recognition peptide that directs the mRNA ribosome complex towards the ER. Here's a picture of that verbiage. What I want you to know, I'll highlight. We'll read through these steps. All right. The mRNA ribosome complex is directed to the rough ER by the SRP. So the SRP, I haven't mentioned that. The SRP is a signal recognition peptide that binds it to a receptor site. So I'll just note the SRP. Sometimes books get too fancy in their talk. All they're trying to tell you is the signal recognition peptide. It guides the newly formed polypeptide into the ER so it can be further processed. That's what this whole figure is trying to show you. Okay. And that actually concludes chapter 3. We're done a little early with lecture today. That's good. What I'd like to do now is take a break.